Hello and welcome to Kernels, a podcast from Ohio Corn and Wheat. On Kernels, we're bringing you updates from your association, your corn checkoff, and your wheat checkoff. I'm your host, Marlene Ike, here at Ohio Corn and Wheat, and I'm joined today by Tad Nicholson, our executive director. He's my co-host today. Tad, welcome back to the podcast. Glad you're here with me.
Thanks, Marlene. Glad to be back. Hey, but today we're talking about vomitoxin and more specifically, really kind of what farmers are asking about, what they're talking about, what they should know about. And we have two very special guests with us, Dr. Pierce Paul. He's the professor and chair of the plant pathology department at Ohio State. And Jason Hartshoe, he's from Crawford County. He's a field specialist for dairy. He's a dairy extension specialist in the state. How appropriate, because the dairy industry, as in all industries, would really like us to figure out some solutions to vomitoxin. It's just a hassle. Gentlemen, thanks for joining us on Kernels.
Yep, thanks for having me.
Thanks
Good.
for having us today.
All right, where are you coming from? Jason, are you coming from Crawford County today, or are you out and about?
I'm stayed in Crawford County today, so I'd be sure to be able to have this discussion with you guys. And it's a little bit wet here today, so planning is waiting a few days to be back in the field. But there is some corn that spiked out of the ground, especially down in the southern part of the county. It seems like they've caught a few less rains than the northern part.
Pierce, where are you coming? Are you in Columbus or Worcester today?
I'm in Worcester today, actually.
All right.
I'm going to head out after this to see what's going on in the field. My guys are trying to get some seed treatment trials planted and get some wheat sprayed. So we'll see.
Wow. Talked to some growers in Southern Ohio today and also in Huron County. And it's everything from we got hammered 2 inches of rain, hardest downpour ever, to all the rain missed us, and we are going hard planting today. So that's the swap. That's Ohio for you.
Yep, pretty much.
It is Ohio. I also feel like it would only be fair then to ask you guys as well. Do you also plant a garden? Are you worried about your tomatoes? Have you planted cold vegetables? What's that like for you guys?
You and I might have to share some garden notes because I'm a very avid gardener. I plant a garden every single year. And I depend heavily on my garden during the spring, late spring and summer months for my actual greens and vegetables. So I do plant a garden, everything from okra, eggplant, tomatoes, butternut squash, and all kinds of different things.
I'm a very basic gardener, and quite often my garden doesn't get planted until June. But the good thing about planting it in June is the garden centers are wanting to get rid of their tomatoes and green peppers and things, so they're on sale.
You get them cheap.
But yeah, tomatoes, green peppers, some green beans, cucumbers, fairly simple. But I do have a garden. It just usually doesn't get planted in a timely fashion.
Yeah, I'm gonna make a shameless propaganda for my graduate students because they do have plant sales. So I buy my plants early to help support them. So if you're really looking for plants for your garden, by all means, Plant Pat Graduate Students Association, you can find some good plants.
Good plug. We need to support the students at Ohio State. Good.
Absolutely.
Good. Well, I'll have to get some advice from you, Dr. Paul, then, especially because we're like in the same zone and have the same garden climate. So I'll have to get some advice. Now, today we're really asking some more hard-hitting questions, though, than probably just gardening advice. Before we jump into vomitoxin and really having you guys answer some questions for that, we wanted to just remind our listeners of what that kind of working relationship is like between Ohio Corn and Wheat and the two of you, and why we're even having this conversation. So, you two are part of a broader research team that Dr. Paul leads at Ohio State, and the Ohio Corn Checkoff has been investing in research about vomitoxin, I think since 2021. So we've had a kind of working relationship with you all with investing in some research, supporting research that you all have been doing at Ohio State, and therefore have been really kind of partners on this project. But it's been a good few years here. So Tad, do you want to kind of give an overview of what that has looked like in the history of the organization and what we're looking forward to moving forward too?
Well, I think most farmers, most listeners are going to remember that 2020 year where we had it statewide. It was just really bad. It was a tough year. So you mentioned 2021. That's when we really kind of got active with Ohio State. Luckily, we've been working with Dr. Paul for a long time on head scab research in wheat. So we do the wheat checkoff and the corn checkoff. For wheat, we've been working on that since the beginning of our wheat checkoff. So a long history on that. And of course, a very close cousin to head scab is vomitoxin and gibberella ear rot. So it was very natural just to call him. We were very fortunate. We have a lot of great expertise and boots on the ground right here to help take care of that, because when 2020 came along, it became very apparent there was no ignoring this anymore. It was happening too often, on a regular basis, all the time. We were going to have to deal with it. So whatever the weather change happened in Ohio, we just own it. And so we're just going to take this on and really try to invest some dollars into trying to figure this out. So we've been actively involved with this since 2021, as you mentioned. But just this year, we've really kind of dove in and really made one of the largest investments that we've ever made in research with our checkoff program. And that's to solve this issue for everybody that grows it and everybody that buys corn wants to see something done about this issue.
So as the year goes on, we're going to have more and more updates. As the next few years go on, we're going to have more and more updates about what's happening with that research and have more of that. That's not really the purpose of today's conversation. So if you're listening going, great, I'm going to get all the answers. We don't have the answers yet, but the research that's being done right now.
We are committing that part of this research is communications. It's going to be going out and saying what we've done, what we know, even if we don't know everything. It's just got 1 year's worth of data. We're not going to hold anything back. We're just going to keep sharing and really hopefully solve some problems as they're happening.
So today we want to dive into some questions that are kind of frequently asked questions about vomitoxin. For growers who maybe have been lucky and haven't experienced it a lot, but likely they may be facing that sometime soon. And we want to give them some just like groundwork and some information. Some of this will be asking a little bit of what we've learned so far. So let's just dive into these. And some of these, I think, Dr. Paul, you're going to answer, and some Jason's going to answer. So let's just start with a really big picture question. And Dr. Paul, I think this one is for you. What is vomitoxin?
All right. So vomitoxin is a mycotoxin produced by several different groups of fungi, and in particular the fungi that infects corn, that's called Fusarium graminearum, produces mycotoxin called dioxinivalinol, and because it causes vomiting in animals, it's called vomitoxin. So vomitoxin is really the common name for a mycotoxin that is dioxinivalinol. Now, what are mycotoxins? Well, fungi produce a wide array of chemical compounds, from enzymes to toxins, that help them to infect plants and colonize and grow in plants or survive in residue in the field. So they produce these toxins for their own benefit. It so happens that some of these toxins are toxic to humans and livestock, can cause illnesses, can even cause death. And when you've got this subgroup of these secondary metabolites or toxins that are toxic to animals and humans, then they're called mycotoxins. So you can find these in a wide array of fungi, but the ones that are toxic to humans and animals are called mycotoxins, of which vomitoxin is one of those.
So when we harvest the corn, that mycotoxin, that vomitoxin, sometimes farmers will just call it vom, but that vomitoxin is going to essentially be part of the grain that's harvested. So Jason, why is vomitoxin a problem then when you haul your grain to an ethanol plant, when you deliver it to an elevator, when you sell it to a feed mill that's making livestock feed? What's the issue there then?
Yeah, so when we think about what the issue is, you say I'm hauling it to an ethanol plant. Well, at first you think, well, that's not going into livestock feed. But the reality is, no matter where we're hauling our grain, somewhere it ends up in livestock feed. And as Dr. Paul mentioned, it causes vomiting at the wrong level, basically, to the livestock, or just complete feed refusal. Swine for Ohio is often thought of as basically the canary in the coal mine, because they can actually, if you would grind feed that is too high a level of DON and you put it into their feeder, they just won't eat it. They might eat enough to vomit once, but then they will quit eating it. And when we think about, ethanol production, so yes, we're getting ethanol that's utilized for fuel, but then you also have that distiller's grain that comes out, which is a very important part of the ethanol process. And that portion ends up going into the feed industry. And when ethanol is made, the vomitoxin is almost tripled during the ethanol process, because none of that toxin actually goes into the ethanol itself. it is all left behind and stays in that distiller's grain that's going to be going to the livestock industry. And, if you deliver to a local elevator that's not grinding that grain themselves, it's going somewhere. And it's going to end up as a food source for something. Even if it's exported, our export market is very concerned about what our vomitoxin levels are, because when they buy that grain, they're buying that grain in order to feed it to something or make it into something else that ends up having a byproduct to go to livestock. And what are those numbers? Why is it so critical? Well, when we think about pigs in general, we'd really like to see a ration under 1 part per million. And the corn going in then maybe can be up to 2 parts per million as we dilute it out with other things like a protein source of soybean meal and other things. And then when we think about high producing dairy cows or really any lactating animal, that 1 part per million is really that ration level we want to stay below, which may allow us to put 2 or 3 parts per million into that ration of the corn coming in or the distiller's grains coming in, depending on what other ingredients we're putting in that might not have any DON in them. But when those animals eat it, there's things that happen before the vomiting. So before that happens, before they get sick, we see a decrease in production. So we're going to see a decrease in the immune system's response. So they're not going to be able to fight off other diseases as well. We're going to see decreased milk production. We're going to see decreased rate of gain. That's really that rate of gain side, what we start thinking about for a flock of sheep you're feeding out or fat steers you're feeding out. Really, that decreased performance starts at about 5 parts per million. Even though we can actually feed a ration up to about 10 parts per million, that decreased performance starts down there around 5 parts per million. And that can be really critical and important when you're working on, making that marginal return on feeding these cattle out. If you're feeding too high a VOM and we lose rate of gain, you may lose that profitability.
So it's really an issue about livestock feed, and remembering that even like no matter where you are selling your grain, there's issues down that chain with livestock feed. So if you're hauling to an ethanol plant, they're still making those, DDGs where it's more concentrated, and so there's concerns, with that. Now, Jason, you mentioned something I just want to confirm for our listeners. I think you said like DON levels and vomitoxin. So confirm for us, when you say DON and we say this is the same thing, right?
And when I'm throwing them back and forth, they're the same thing. Vomitoxin is somewhat a class, but here in Ohio, when we're talking about vomitoxin, we're really talking about DON. There's actually a couple other toxins I won't get us down into the weeds on that same Fusarium granulorum that Dr. Paul mentioned actually produce that causes a whole different set of issues in the livestock industry. But we won't get down into those weeds today, as our primary challenge comes from the DON portion normally.
Yeah. So as an Ohio farmer, when you're hearing those terms, we're really talking about we're using them interchangeably, even though in a very scientific sense, there's some distinctions there. Yeah.
So DON is the short for dioxynivalenol. So that's what I said. Vomitoxin is the common name for dioxynivalenol, and then DON is the short name. So we got to be careful. But when we're talking DON or VOM or vomitoxin, we're talking about the same thing. It's correct.
So Dr. Paul, when does vomitoxin occur? When does that become a problem in the production, like the corn production cycle? What month or what cycle? When is that happening in the course of the year?
Right. So vomitoxin or DON production does not occur in isolation. It has to occur as a result of fungal infection of the corn ears. In other words, the fungus has to get into that corn ear and start growing, and then when it grows, it produces DON. Because what DON does is DON kills the plant tissue ahead of the growing fungal mycelium. So unless that fungus is in there and growing, DON will not be produced. So when does that infection occur? Infection occurs during that silking growth stage, during that week to week and a half after silking, because it occurs directly via the silk. That's where natural infection occurs. So when you've got ears developing and weather conditions are favorable and production conditions are favorable, that fungus enters the ear via the silk. And once it enters the ear via the silk, it starts growing. And as long as it keeps growing, weather conditions are favorable, hybrid is susceptible, fungicide is not applied. Once it keeps growing, it's going to produce vomitoxin. However, if you've got damage to the ear by birds and insects, that fungus can get in via wounds or damage as well. So it doesn't have to necessarily wait for that that silking window, that R1 growth stage. It can occur if there is damage to the ears by insect, by hail, by birds. The fungus can get in there as well.
And I know we're going to get into some of like what are all the different factors that a farmer could manage to try to mitigate that and minimize it. But you know, Tad, I've heard you talk about how you've heard of farmers based on planting date, because this has to do with silking, which is related, of course, to when you plant. But you could have a cornfield on one side of the road and another, and one has an issue with vomitoxin and one doesn't. And it might be because of planting date as well. So you know of differences there and have heard things.
Yeah, you've seen it. And that's what really causes a lot of mystery around that. Farmers would be so frustrated because I have the same variety of corn planted on 2 fields across from each other. It's just that one was planted 2 or 3 weeks later than the other one, and one has vomit and the other one doesn't. So it definitely is. And it kind of goes back to what Dr. Paul was talking about the when silking is a very important time of year for us. So that's what we're all focused on.
Planting date doesn't tell the whole story. Planting date is actually a surrogate for favorable weather conditions at silking. So planting date could cause silking to be a week earlier, a week late. And that week earlier, a week late makes the difference in terms of conditions being favorable for that fungus to infect via the silks.
Yeah, you see those times when you would put the plant in the ground and it would turn cold, wet, and it just takes forever to get out. That planting date's very different than something like this year where you've seen it pop out of the ground really quick after planting.
So Dr. Paul, again, there's a lot of factors there, but can a farmer plan and prepare for possibly having, vomitoxin in their corn crop? Is that something that you can somehow prepare ahead of time for?
Yeah, before I get to that, let me talk a little quickly about can we predict vomitoxin? And the answer is yes, we can predict the risk of vomitoxin contamination. Because like I said before, vomitoxin contamination doesn't occur by itself. It's associated with fungal infection. And for infection and fungal infection to occur and for that fungus to grow within the ear, it requires specific environmental conditions. And Todd mentioned support that we got from the checkoff, which we're very grateful for. But he forgot that prior to the 2020 year, we've got some partial support from the board to look at environmental conditions that drive vomitoxin contamination of the grain. And from that project, we understand the window during that growth stage, during our silking growth stage, and the specific weather conditions, combinations of temperature and relative humidity. Once we understand what are the combinations of temperature, relative humidity, hybrid susceptibility, cropping practices that drive vomitoxin contamination, then we can use that to predict. So yes, we can definitely predict vomitoxin contamination because we've done a fairly decent job of understanding what are the specific temperature requirements, rainfall requirements, relative humidity requirements on a given set of cropping practices that can drive down contamination. If farmers understand and use these tools that we're developing, then they can prepare for vomitoxin contamination. I think the first line of preparation would be selecting hybrids. And I know Jason's going to talk a little bit about that, but you can select your best resistant hybrids. And that's a good place to start. Select your best resistant hybrids to plant because not all hybrids are made equal when it comes to vomitoxin contamination. Some are more resistant, some are more susceptible. Seed catalogs can be used to get an idea of which hybrids are more susceptible, so you can avoid those. That's a good place to start. You can also do some form of crop rotation or tillage because the fungus that causes vomitoxin survives in the stubble in the field. So if you get rid of some of that stubble by tillage to bury it, that's going to speed up decomposition. Or you rotate away from corn or wheat, which are hosts for the fungus. You can help reduce the risk of vomitoxin contamination. And then once you get into the season, you can use a tool like a forecasting system to say, hey, the risk of vomitoxin contamination may be high. I can potentially apply a fungicide to reduce that problem.
So Jason, let's go right to that question that Dr. Paul kind of teed up for you. Talk to us about selection of the hybrid you want to use, and does that change the susceptibility?
Yeah, so that's a really important part. Really, everything in corn production starts with hybrid selection. When we're trying to figure out what we want to plant on which field and which conditions, which hybrid is going to do the best on those soil types, we spend a lot of time picking out those hybrids for their agronomic factors and for their disease resistance factors and things. And the same thing comes into play on the Don side. There's a big difference in hybrids. Unfortunately, a lot of seed companies don't have a really strong published rating system yet. And a lot of that's because this isn't a nationwide problem. The whole country isn't dealing with it. So when they look at their hybrids and rating them for diseases, they look for that wide area that they're going to be screening hybrids for, and they select for those different diseases. And really, this is more of a Great Lakes type issue that we're dealing with here, just due to those weather conditions that drive disease development. So we don't see as much in the seed catalogs, but there's different things you can do. One is, tracking your own hybrids and how they perform on your farm. Now you really want to go back and look at those weather conditions. It's kind of one of those things where if you have a high hybrid, that tells you it's susceptible. But if you have a low hybrid, that doesn't tell you it's resistant. What that tells you is you didn't have favorable conditions for disease development during that pollination window, or there was no insect feeding or anything that allowed for fungal infection to occur. So we're really looking, at needing multiple years of data going into a hybrid to say if it's susceptible or resistant. But thanks to a project that was actually funded by Corn Board, we kind of started looking harder at that hybrid resistance and plan to continue to do that. And some of that's going to be to actually identify better identify the characteristics of those resistant hybrids. But this past year, we had 3 different locations planted to about 80 hybrids. And we harvested those and collected Don samples. Those were all inoculated to try to guarantee disease. And out of those 80, we had 13 of them that across all 3 locations averaged less than 1 part per million. The range on those was about 0 to 2.4. So just because it was below 1 as an average, there had been a plot maybe that was a 2.4 parts per million. So there is, some variation there. But then when we look a little further, there was 28 hybrids that averaged below 2 parts per million, with their highest plot being 4 parts per million across that project. And then when we start looking at 3, there's 43. So half of those hybrids across all 3 locations averaged less than 3 parts per million. So then the highest hybrid was somewhere around at one location was just under 30 parts per million, and there were zeros on that same location. Now that was across the maturity spread, but when we looked at maturity spread, we would have had favorable conditions across all of our pollinations, because across our maturity spread, we had even disease development. We had some susceptible or high Don hybrids on the short maturities and in the long maturities in the medium groups. So we had those favorable conditions showing that there are those differences between hybrids that we really need to continue to investigate, because that's really going to be the first step. You can't make a really susceptible hybrid low just by putting a fungicide on or doing the best management practices, unfortunately, if we have favorable disease conditions. But some of those really good hybrids are also, some of the company's favorites because they yield really well and perform really well or have good disease packages for other areas of the country. They just have a weak spot, unfortunately, for us here in Ohio.
Well, let's touch on that really quickly. And Tad, I want to go to you for that, because you are often working with your counterparts, so the executive directors of other state corn grower associations throughout the Midwest and the country. And this isn't an issue everywhere. And so as companies are looking at their, what hybrids they do make available, of course, they've got to look at what they need across the whole board for that. And so there's a little bit of an issue there too, and that it's such a regionalized problem for us.
Yeah, so my colleagues and I that do the same job all across the Midwest, we meet at least every other week. And I can remember vividly in 2020, 2021, right when we realized, gosh, we're in trouble here. We got to do something. We're pulling together this team that led by Dr. Paul. All of Ohio State was asking, where are our resources? I got on one of those calls and said, look, Vomitoxin it's a tough problem for us. Who wants to work with us on this? And I heard crickets on the phone. It was like we were all alone. There was this wall around Ohio that year that said, no one else has this problem. It's all yours. And so this past year, we did get some attention from Michigan, Indiana, but really it didn't go much further west than that. And so I made some jokes before, and so I'll just clarify it. I'm just kidding on this, but I did say, look, if we really want to get the seed company's attention on this, we'll take some of these spores over to Iowa and we'll just sprinkle them around a little bit over there, and then I think we'll get everybody's attention on it. And I told my colleague Craig Floss, I'm just kidding. If you do get a vomitoxin, it's not me. But it has been very isolating here. It was like, no one wanted to work with us. But this past year, we've seen a lot different picture. And so I think Ohio State is really in a position of leadership on this. Everyone's sort of watching us, seeing what we're doing, and want to try to amplify what they've started here.
Yeah. So there could be, there might be some different changes we see there in how companies are operating as this potentially becomes wider spread. But if you're an Ohio farmer and you visit with your friends around the country, you've probably already realized like this has been a little bit more local to Ohio and hasn't been something that farmers across the Midwest are facing yet. But Dr. Paul,
Marlene, yeah. I just need to add too, one of the things that we've noted is Ohio State doing these variety trials. It's the only place that's happening as a third party. Some seed companies are doing their own kind of evaluation of their own, but to have a university land grant doing that, this is the only place that's happening. So I think it's really a very important thing because it causes some great conversations among plant breeders and the faculty like Dr. Paul and Jason, just about some of the things that they've noted in here. And I think there's some great progress that can be made on that. So I'm really proud of that. This is only 1 year into that, and I hope it's several years into the future.
So Dr. Paul, switching gears, besides hybrid selection, what impact do other practices have on maybe preventing that? Or does there's a lot of things we want to be doing in Ohio related to H2 Ohio funding and just climate smart agriculture in general, like no-till and cover crops and those types of things. How do other practices play into preventing vomitoxin?
Right. So hybrid selection is the first line of defense against it for sure. But like Jason said, no hybrid is 100% resistant. So you're still going to have some contamination if you don't use a wide range of approaches that we typically call an integrated approach. In addition to using your best resistant hybrid, you probably want to do some form of crop rotation. Rotate away from corn or wheat. You probably don't want to follow corn after corn or corn after wheat in successive years because that fungus survives in that crop residue. In that crop stubble, it's the vomitoxin and the fungal survival is much greater in residue than it is in the grain itself. So if you plant corn after corn, that fungus is going to build up in that field over time. So you really want to do some form of rotation. If you don't have the option of rotating, then you probably want to do some form of minimum tillage. We know of all the advantages of no-till, but one of the disadvantages of no-till is fungal buildup in the soil. And if that fungus build up in the crop residue, then you're going to have a higher risk of vomitoxin contamination. So if you can rotate, some form of tillage to get rid of that crop residue. I know of no specific example of cover crop affecting vomitoxin contamination, but if you've got a grassy host as your cover crop, this Fusarium graminearum, the last name graminearum, means of the grasses. So if you've got a grassy species, it's possible that that fungus can survive because it not only affects corn and wheat and cultivated crops, it can affect grasses as well. But I know of no specific example where cover crop actually increased the risk of vomitoxin contamination.
So how about fungicides? Do fungicides work? So if you know you're at risk or you have something you've observed in terms of your risk, do those work? How are they best applied?
Fungicide can be effective. We're still doing a lot of fungicide efficacy work for this disease. Unlike Todd mentioned scab earlier, we've done more than 20 years of fungicide efficacy research in scab. We've made tremendous progress in terms of what to apply, when to apply, and how to apply. And we're going through the same sequence with some funding from the board as well to understand which fungicide to apply, how to apply, and when to apply in corn. Fungicides do work. We've got, you have to choose the right product. You have to apply it at the right time, which is that R1 that silking growth stage, because remember, I said that's when the fungus infects. So you really want to put on that fungicide just about when that fungus is about to infect. And then you want to apply it with the right method. If you think about applying a fungicide to a corn plant, if you spray over the top, most of that fungicide is being held on those upper leaves. They're not getting down into that corn crop to the silks. They're not getting down into the canopy. So the right application method to get as much of that fungicide into the canopy as possible to protect those ears would be the best approach. So the right product, the right time, and then apply the right method. We're playing around with some drone application methods. We're looking at some different nozzle configurations. We're looking at different volumes to see how best we can get that fungicide into that canopy, close to that ear or on those ears to provide the best protection. We're getting some good results, and we're going to continue that research over the next year or so as well.
You know, just on that note, this is one of the things that I really hope there's some great discovery on this, because it makes logical sense as he described that aerial application is going to be very challenging, but it's also the fastest, and the window to get this done is a very small window of opportunity. So if we can't aerial apply it, there's just only so many ground rigs that have wide drops that are going down through you know, standing corn, and so that is it's a slower way to get things done, but it is a very important part of this research will be how can we most efficiently get this fungicide applied. There's an economics thing to this too.
Absolutely.
And so look at this year right now, this winter, farmers were doing all their, you know, Barry Ward's, you know, his budgets, and they're looking at this at some either red numbers or some very close to break even numbers. So if you're making a tough decision about what to invest into your crop, fungicide might be one of those things that you decide this year, let's just not invest in that. It's too tight of a year. Well, if we get caught and we have an idea that comes silking that this is going to be something that's that you are in trouble at the end of this year, you're going to be scurrying around trying to figure out the fastest way to get these fungicides applied. So this is going to be a really important part of our research that I'm really looking forward to sharing with all of our members.
Mm-hmm. You know, like I said before, and Jason mentioned this too, fungicide alone is not going to do the trick. We see the best results with fungicide when we apply them to our most resistant varieties. So you still got to choose resistance, and then the fungicide, even with the aerial application, is going to provide some complement. You have to help that fungicide to be effective because of the challenges of getting it on the target.
Well, like you said, Dr. Paul, it's really that integrated approach, right?
Absolutely.
Where you've got it. There's all these different facets of it. Really diligent in being smart about all the different facets of it and making good choices and management decisions along the way. And we're really excited about what the research are doing right now. Like Ted said, there could be some really great discoveries that come about this. But right now, and sometimes, no matter how great of management decisions you make, you're still going to have corn that's going to test positive for vomitoxin at the end of the year. So Jason, a couple questions then for you. What can farmers do with corn that's testing positive for vomitoxin? Kind of walk us through that. Some farmers have been experiencing that firsthand, and some maybe haven't had that pleasure yet.
If you're in Ohio and you haven't had that pleasure yet of having a high vomitoxin test, consider yourself to be very, very, very lucky. Because just about everybody by now across the state has experienced that at least once, where they pulled up to the probe and they found out that their load was either high enough they were going to be docked or high enough they were going to be rejected. And at that point, you know, what do? The management that Dr. Paul already discussed, you know, hybrid selections out the window, fungicide applications out the window, tillage to limit spores so we don't get that infection, all of that is gone. So now what do we do that we have this grain? And really it starts with knowing your numbers at harvest. So if you have on-farm storage, you really need to know the numbers of the corn you're harvesting and putting into your own bins, because that's really where it starts, when we can start making some management decisions and doing things differently. If we know we have issues before harvest even begins, that first step is to take those fields that were at the greatest risk and potentially harvest them a little bit early. Yes, we may be talking about harvesting them close to 30% moisture, or maybe more like 27 or so. So they're not quite as soft and we don't have as many combine issues. But when we harvest early, we decrease the amount of growth of that toxin within the field. The longer a corn hybrid is in the field after it's been infected, the more time that fungus has to grow on that ear. So the higher those DON numbers you get. That's why often we may start the season with really low DON numbers. And then by the time the season comes to an end, we have those high numbers. It's not that that infection occurred during, that harvest dry down. It's that the fungus continued to grow and continued to produce DON. So really that first step is some scouting pre-harvest to know if we have a problem, and then picking those fields or those hybrids that are at the highest risk to go out and harvest as higher moisture corn and spend a little extra on dryer gas. Now, during that harvest process, and one of the reasons why I say it's really important that you know what's going on in your field as you harvest, what your DON numbers are, is because there's a few things we can do at the combine. Anything we do to try to blow fines out and to do a better job of not breaking cobs on the combine will actually lower those DON numbers a little bit. So the cob often actually is really high because not only does it a kernel infection, it'll actually infect the cob also and cause a cob rot. So that cob then is more brittle. So when that breaks, not only does it leave dust, but there's also little pieces of cob in there that can get picked up and ground up in the test that can cause problems in those high DON numbers. And that high DON out of the cob, that's real. That's going to be going into the feed stuff in the end. So anything you send to the elevator eventually is going to end up in feedstuffs. So you want to be careful. And that's one of those steps, is to try to keep as much cob and fines out. So setting that combine a little bit better so we're not breaking cobs, and potentially setting your screens on the combine, on the cleaning sieves, so that we're getting as much air as possible through to blow as many fines out, broken kernels and things out the back of the combine, and leave that in the field. Unfortunately, that's leaving inoculum for next year's crop that maybe we want to go back to some of Dr. Paul's other IPM on, but that's kind of like step one. And then from there, when we know we have high DON, we take it into our facility, we dry it, and then we can start to consider some other things. So the first thing to consider really is, how do we store that corn? So we want to make sure we get that corn dry, below 15%. Ideally, if we're going to store it for multiple months, this is probably the grain we want to dry closer to that 13 and a half, a little bit below trade moisture, so that we're sure all the grain is below 15%. So we don't have increased growth of that DON fungus within the bin. But when we think about that increased growth, it comes down to bin management. We have to make sure we get that corn cooled properly, ideally down around the mid 30s, so 35, 36 degrees. When we have those cold nights, that's actually a good crop to try to get all the way down to 32 and hold it there as cold corn so we don't have fungal growth in it, and make sure that it's evenly spread and we got those fines out pre-drying so we don't end up with a wet spot or a hot pocket within that bin. Because those are all areas that are going to see actual DON growth within storage. But if we do storage properly, we won't have an increase after it goes into the bin for storage. So I talked about getting those fines out. So one of the other projects we've been working on and really continue to plan to dive into, worked with a few growers on, utilizing some of the funding from Corn Board, is to look at grain cleaning to lower VOM levels. We're still working on really being able to quantify what different cleaners do. But across the ones we've worked with, we've seen that the more fines we get out, will lower that DON level by a few parts per million. But it's also important to get the overages out, or the cob. So we need a cleaner that does that's very effective on both sides at removing that cob and removing the fines, really to be able to get that lowest DON level possible. And it's not going to take a 13 down to a 2. It might take a 13 to an 11, or a 13, down into the single digits. But it's not a miracle cure, but it is helpful. And every little bit that's removed helps.
So Jason, it's hot off the press. You haven't seen this yet, but we just got our first set of data back on the cleaning. And you're right, it reduces vomitoxin in the grain after cleaning by about 12 to 16%. So that's, for me, it's a significant reduction. And the screens, the fines, had vomitoxin levels in the upper 20s and low 30s, whereas the grain itself had vomitoxin in the single digits. So it's clear that if we can get rid of those fines, like you said, we can reduce the overall vomitoxin level. It is a complementary approach to whatever we do in the field, but at this stage of the game, whatever we can do to reduce vomitoxin, I would definitely take a 4 to 16% reduction or 12% reduction after doing everything we do in the field.
Yeah, if you have a bin full of corn that nobody's interested in buying, it's worth the hassle of going through that. Although I think everybody agrees, and our checkoff board too agreed, everything that we want to do in research is how do you keep it from getting to this problem in the first place. But if you've got that problem, you got to deal with it because there's no one else that wants to come and help you with it. So good job on that research, Jason.
Yeah, and that's important. So one thing I guess I didn't touch on back on the elevator section, you know, a lot of that grain when we deliver it to a local elevator, I mentioned it ends up in feed products. Well, a lot of our grain that goes to our local elevator may actually be going into leaving Ohio's pork industry, but moving into the pork industry on the East Coast. And one of the challenges they run into is our rail system is really designed to move our grain from Ohio to that section of the industry for the pork industry. Well, when we have high vom corn, there's a home for it as long as it's moderate vom corn to go mostly into the poultry industry, which often means it needs shipped down more into the Gulf area. And for them to rail clean corn out of the I-states, which usually send their corn to the Gulf, to the Carolinas, and for our corn to go down to the Gulf instead of the Carolinas, causes this major logistic shift requirement that is not easy for our end buyers that are going to be using that corn and in the rail industry. Because if we look at a rail map, one railroad does not actually go everywhere in the country. There's different companies and they really don't, you know, their lines don't go the same places. So they have to figure out on that shipping, potentially how to move from one rail company to the next, which increases their expense on getting that grain to market.
Raises the price of everything for sure. And it's an expensive issue, you know, all around. We think about how farmers are managing it, how our end users are having to deal with that. So Dr. Paul, we're all really excited about this research program that you're leading that includes several projects that you've been doing and for the next few years of research. And a lot of the discussion has been, how do we minimize the risk? So the last question I really want to ask you is, long term, in a perfect world, do we think that there will be a time where we could eliminate vomitoxin completely, or is this always going to be a discussion about just minimizing it and managing it well?
I think ideally it would be great to be able to eliminate vomitoxin completely, but given what I understand of diseases and how they develop, using scab as an example, it's going to be very difficult to eliminate it completely. But what we're going to do is learn to manage it and learn to live with it. I think as we do some of this research we're doing, understand hybrid reaction to the vomitoxin contamination, select hybrid that are resistant. Part of the research we're doing is what we call characterized resistance. If we can understand what it is about a certain hybrid that makes it more resistant to another hybrid that makes it more susceptible, then we can provide seed companies or breeders with that information so they can breed those traits, genes, into hybrids, so you have more and more hybrids being commercialized that are resistant. But you need to understand what makes a hybrid resistant and what makes another hybrid susceptible. And part of the research we're doing with some very meticulous work done by a graduate student to understand and characterize resistance. And once we have that information, we're going to share it. But the long-term goal is to get more resistant hybrids out there. And if we don't eliminate it, we're going to get it to a very, very low level long-term and learn to live with it.
Well, I feel like the moral of the story is stay tuned for the discoveries that we're going to be finding out that you're going to be sharing with us as a result of all this research. Yeah. So again, the corn checkoff is investing heavily in this area. As Tad referenced I think towards the beginning of our conversation, this is the largest research related investment the corn checkoff has made in the history of the corn checkoff here in Ohio. Now, Tad, I know you're kind of the co-host today, but you have to answer a few questions too. So we want to, before we wrap up, we want to just have Tad address a few things, because we've been talking about the research side and what the checkoff has been doing. But there's also some other pieces here that our organization is working on. So for example, I know people, they'll pull into an elevator, they'll test high enough that load gets rejected, and we know that folks are just pulling out of the elevator, driving around the block, and getting in line again, and then that load passes. How can we kind of work on that issue about how the testing functions?
Well, this is one of those wicked problems that we've really, I have to admit, we have not been able to crack this one. We studied this we create these strike teams in Ohio Corn and Wheat to really dive into an issue. And we did that back in 2020 when we were dealing with this so much. And that strike team went the entire swath of all of the research that we've been talking about, what can be done, before you got it, what can be done after you've got it, what's the testing protocol, and how are these done? How can we improve that process? And that testing is really the one that we've had the biggest problem with. And it's because it's not really about the sophistication of the elevator, from a country elevator to a big, ADM or a Cargill type of facility that has more sophisticated equipment. It's still, you see those examples of, it's the luck of where that probe goes in the truck. And it could very well, and it's also just about the suction of it. Did it go all the way to the bottom? It's a luck of the draw type of a test oftentimes. And I don't know how to really change that, because what are you going to do? Take 40 probe samples out of a single truck? That's the only way to really get a more representative example of that. We've had some other problems. This is real life scenario, and it has to do with crop insurance. So if you have Vomitoxin and you go to the elevator and they say, we're not interested in having this, or we have a massive dockage of the price of your corn. So you go and you call crop insurance, and they come out and they have to have a sample taken to a federal lab. So they probe your bin. Well, if probing a truck is the luck of where the probe is stuck, what do you think that probing an entire grain bin is like, and how to really get a representative sample there? But that's what crop insurance requires. So this is real life. You go to the elevator, you have high vomitoxin. They say you got huge dockage. You call crop insurance. They come out, probe your bin, send it in, and they say you only have 1 part per million, so no crop insurance. And so that problem, we've talked to RMA about. We've been in touch with them to talk about ways that we could strategize on that one. But honestly, that one has not been cracked yet, but certainly has not lost on us, because every board member that we have has dealt with this same issue that every one of our listeners are, that can relate to this scenario. They've all experienced that, and that frustration is felt by everyone. So we're continuing to work on that one. Haven't cracked that one yet.
And we're not working on this alone. I mean, we mentioned before, this has kind of been an Ohio or Great Lakes area issue, but and while Ohio State has been kind of leading the pack on the research on this, we're not the only ones concerned about this. So Tad, who are the other organizations and companies that we are, trying to kind of pull together in a collaborative effort?
Well, like I said, I felt like we were all alone in this, and there wasn't anybody else who wanted to really work with us on this. But Jason and I got involved with a meeting that was really convened by Beck's Hybrids, and I got to hand it to them as a seed company. They have enough people talking about this, and they also talk to people like the North American Millers Association. So the ADMs and the Cargills and the Bungies all belong to North American Millers Association, and they've been mentioning this at their various meetings. So it's got the attention of the North American Millers Association, the American Seed Trade Association, which Bex is involved with, brings about, this conversation as well. So all of a sudden, Kayla Jenkins over at Bex, got to hand it to her. Anybody working with Bex, she's doing a heck of a job over there. She convened a group of maybe 30 people, Jason. I mean, it was anywhere from you and your counterparts at Purdue. Michigan State wasn't there, but they had some growers from Michigan that were there. But all of those universities that really want to work together on this, in addition to the North American Millers and ASTA, and various growers around that. I mean, it was a lot more people that really are interested in this than I had initially thought. So it was really great that Jason was able to be there and describe what we're doing, because also the Indiana corn checkoff and Michigan corn checkoff are partners, and they have the ability to influence this as well. So we're not alone anymore, Marlene, and that's a good thing.
Yeah.
That we're not as lonely as I thought we were. But it certainly is something that's gained a lot of attention, and it's going to keep on. So this topic is going to be brought up at the national meetings of the Seed Trade Association and the North American Millers. We get this inserted into those national conversations, and I think we make good progress.
Well, I've really enjoyed our discussion today, and I hope, dear listeners, I hope that you have just learned some things maybe about Vomitoxin you didn't know before. You had some questions answered. I hope also that in the coming months and few years here, when you see Dr. Pierce Paul and Jason Hartshue's name on extension programming or anything like that, that your ears really perk up and you're paying attention as they really have a chance. As they're sharing about the research too.
And Marlene, I just can't say how lucky we are to have these two. But this research that we are going to be embarking on are going to allow them to expand their team. So there's postdocs, there's grad students, there's interns that are all going to be part of their small army of people working on this issue. And all of those, as they graduate, are going to have this knowledge as they go out into the workforce, and they're going to bring this knowledge about Vomitoxin and research with them out there. So I think this is a really great thing. It's a good investment by our checkoff. When you have a corn checkoff like this, when I'm done with my career, when Pierce and Jason are done with their careers, you're going to look back on something and think, did I do anything that was really worthwhile? And if we can have an impact on solving this wicked problem that is such an economic impact for everybody that grows corn and everybody that buys corn, I would feel really good about some of the work that we're doing today. So I'm really anxious to share this as we go along, but we're just at the beginning of what could be a really cool research project.
Dear farmers, if you ever wonder if your corn checkoff is doing anything for you, this is like a great example of something that you're likely facing. And your checkoff was invested in research to make a real difference. So Dr. Pierce Paul, Mr. Jason Hartshue, thank you for being great partners. Thanks for being on the podcast today. If we get more kind of listener questions, we'll feed those back to you. And we're also just really anxious to continue this conversation and bringing updates to folk as the research continues to unfold. So thanks for being with us on today's episode.
Thanks for having us.
Thanks for having us. It was a pleasure.
Well, that's all for this episode of Kernels, folks. Share this with a friend who you think needs to hear about Vomitoxin. Share this if you learned something today. We know that more people find out about the podcast and about these conversations if you're willing to kind of just do word of mouth and share these type of episodes on social media channels or text these links to your friends. So make sure to rate and review this show so more farmers can hear about those topics that matter to them. As always, you can catch us in between episodes at ohiocornandwheat.org or on your favorite social media channel like Facebook, Twitter, Instagram, TikTok. As always, thanks for listening, and we'll see you next time.